Director, Ministry of Health in Oman. So, please.
Good afternoon. Many thanks for organizers for this invitation to speak. We are part of Ministry of Health, National Genetic Center, and the Genetic Center was built in order to provide medical services in Oman with newest genetic technology. The mission of Genetic Center is satisfy the needs of Ministry of Health and Omani community for genetic services and apply newest genetic technology to maintain high quality medical care. So the drivers for the change and application of genetics were brought by improvement of clinical services, increase in survival of the affected by genetic disease, escalating needs for genetic tests and greater diagnostic and care possibilities, and also support from emotional and otherwise support from international collaborators like the World Health Organization, World Alliance for Prevention of Genetic Conditions, March of Dimes, and many others. We have few goals and improve care of people in Oman and development of preventive strategies and build our capacity and use advantages in genetic technology in providing better services, and also update genetic professionals and empower Omanis with the knowledge how to avoid genetic disease. So the sustainable technological capacity is needed for development of genetic services, and we had a lot of help from Western countries in technology transfer. We had visiting consultants, we sent our people for training, we've been involved in collaborative studies, and now we are embarked on international accreditation procedures. I will take you through the steps just to explain you what it is. So first we developed karyotyping, and this is a complex technology when you grow white blood cells and then examine chromosomes, and that was, we trained people in Germany and UK for that, and we had a lot of support. So genetic imbalances cause disease, like you can see this is normal chromosomes of the humans, and if there is 3 copies of the chromosome 21, it causes Down syndrome. The Down syndrome rate in Gulf countries and in Oman is extremely high. It's 1 in 350 deliveries, and even a young woman are at risk. So if you go to handicapped schools all over Oman, you see classes of Down syndrome children, and maybe 20% of handicapped in those schools are Down syndrome. Our scientists uptake this PhD study in Berlin University looking for genetic cause of non-disjunction causing Down syndrome, but it was not there. So we can think that perhaps environmental or other factors play a role. We had mastered another technology, which is looking at the micro parts of the chromosomes, and this technology called FISH, fluorescent in situ hybridization, where you put fluorescent probes on the chromosomes and you can see if the gene is there. For example, here you see the marker for the chromosome, and the red signal is absent on the second chromosomes, means the gene is deleted. The next technology we mastered, it's arrays. So if a normal karyotyping resolution, you can detect 5 megabytes of DNA, and next in the arrays you can DNA, and you can examine every 100 kilobase, and in oligosNP you can go to 10 kilobase, which is very small structures. You can see if they're responsible for disease. So these arrays, you look at the chromosomes, and then if you have any disbalance, you can look in details, and then this part can be also multiplied, like so to look which is the gene involved, and then you look in the databases. So this technology was supported from Lund University in Sweden. And they're still consulting and helping us in complex cases. So this is like just explaining and saying, so this area is abnormal, and then you can see here that things are missing, and you go on to see what genes are involved. So the next step to examine genetic basis of disease in more detail is to embark on molecular studies, trying to see if there is any change in single base pattern. And still, this is in process of development. We have a sequencer, but as technology moved, you can imagine that Human Genome Project, which was costed $30 billion, now you can do the same for almost $3,000, and these machines become widely available. They are widely available. Only the technology is quite complex, and it involves bioinformatics. So we are working on this now. These technologies are all can be utilized in national projects. For example, hemoglobin disorders are very common in the Gulf and in Africa. And it's not to be seen as a defect, because the malaria was pandemic in this area, and the carriers of these disorders had advantage survival in malarial areas. So we developed a lab for looking at molecular basis of this disease with the help of Oxford unit in UK and Leiden University in Holland, and also Heidelberg University in Germany. So we also have a national service for cancer care, and it looks now that hematological cancers, if you know the genetic basis of the malignancy, you can offer better treatment, and you can make the life expectancy much longer, and curative rate is much bigger. It moves from 30 to 90%, the success rate. So this is also very complex technology, and it was supported from a few universities in Germany and in Holland. So this is a cancer registry of Ministry of Health. You can see in females, the thyroid and breast cancer are the highest rate, and in men, the lung, the stomach, and the prostate are the highest on the list. So this push us to develop services, and we're developing breast cancer services in Oman. Dr. Nishat is here. She's developing this service, and we're also developing colon cancer services, and we also look for help and support in universities. We have collaborators in Holland, in Germany, and UK. So what happened in Oman, that the infant mortality rate dropped from almost 100 to 6, and it is all data 2005. Now it's about 7. And before that, in 70s, it was 200. So 200 children born used to die per 1,000 from perhaps infections. But now, as infections disappeared, more children survive. But some survive with disabilities. And for example, this child is mentally retarded with small head. And Harvard University helped us in diagnosis of these children. It's MPCH3 and 5 mutations, which are in Oman responsible for this condition. So I give you a few examples of the studies we have done. This also Harvard University helped us study the families with Cohen syndrome. It's also microcephaly and mental retardation. We had families which many children had a birth asphyxia, but they never had actual asphyxia, but they had the same type of cerebral palsy. And Harvard University neurology discovered that they have a mutation in SPG, spastic paraplegia gene. Families like this, we see continuously all children from one couple, and this is a new mutation of SOX S gene on chromosome 12, which was detected in Germany. Families also exist where children normal socially, but they cannot learn, and it is non-syndromic mental retardation, and we have also new gene for this worked up at Max Planck in Germany. Now in Weill Cornell in New York, under study this family where 8 children suffer from extensive brain calcifications, and they sort of degrade very early in life, and there is a new locus on chromosome 2. There were a request from Germany, our collaborators, to give us help in finding children with wrinkly skin syndrome. They had 3 children in Germany, 2 in Germany and 1 in Belgium, and they couldn't complete the study. So we found 17 children from Oman, and the gene was mapped, and it's responsible for glucosylation of the skin, in Golgi apparatus of the skin. Recently, we had answer to this condition, which is lipodystrophy, but children suffer from cardiac arrhythmias and long QT syndrome. They may die suddenly, but it's also generalized lipodystrophy. And here, a new cavin abnormality of caveola was detected, and it was fully worked up how it works, the disease. And perhaps now they will find some possible cures for it. Robinov syndrome is also a condition which was very rare somewhere, but present in Oman commonly. Very short children with skeletal abnormalities, and it's university in London, St. George's Hospital Medical School, who done the job. And we have another one. This is in France. This is Schwarz-Jampel syndrome, and 3M dwarfism, which was recently completed in multi-center study in France. Children don't grow, they're extremely small, and this is OBSL1 mutations. So I can show you so many of these examples. For these conditions, for example, the treatment is now available because they knew the chemistry of the gene. And currently, we are working very hard on accreditation procedures with help from Lund University in Sweden. So in the end, I can say that collaborators contributed to our technology transfer, to improving services and training of Omani professionals and research. And there were quite a few countries in Europe and USA, quite a few universities who done it on research basis, not asking for anything in return, and we are very thankful to them, because these services are very important for Oman in reducing handicap, improving care. So we looking into a future, and we want to prioritize the state-of-the-art genetic services here, and we have a lot of collaborators abroad. Thank you very much.
Guido Zebic from